This is again, phase 2 randomized trials where a patient are either randomized to standard care high-dose interleukin-2 or high-dose interleukin-2 plus entinostat. Entinostat is a small molecule, is a pill the patient take every 7 days, along with the high-dose interleukin-2 that is administered intravenously 5 days on with 1 week break and then 5 days on. And this is actually has been repeated up to 3 times, so that depending on the response of the tumor. And this study is targeted to a specific group of patients with the clear cells or predominant clear cells renal cell carcinoma who are fit to receive high-dose interleukin-2, which is a regimen that's been approved several years ago. The 2 main really therapies available for this disease are we call a receptor tyrosine kinase inhibitors like sunitinib and pazopanib. These are drugs that are meant to inhibit tumor angiogenesis, the new blood vessel formation, but we know that to have also a direct effect on tumor cells. In first-line therapy setting, for selected patient population, as I mentioned, we still offer high-dose interleukin-2 because that's been proved to have, in a small number of patients, a significant clinical benefit and duration of response. This study is actually a continuation of prior studies. We show that this combination of entinostat, that is histone deacetylase inhibitors, in combination again with the high-dose interleukin-2, is really compare favorably with historical data of high-dose interleukin-2 alone. So based on these initial positive results are now try to confirm this hypothesis that indeed the entinostat may increase the clinical benefit of patients receiving high-dose interleukin-2. And this is also based on preliminary data that suggests that, again, with modulating the immune system with histone deacetylase inhibitors, we can increase the efficacy of immunotherapies, not just in kidney cancer, but also in other tumors.
The purpose of this study is to compare any good and bad effects of using high-dose interleukin-2 and entinostat to using interleukin-2 alone in advanced renal cell carcinoma, a type of kidney cancer. The usual approach to treating this type of cancer is to remove the kidney or use therapies that target the growth of blood vessels or certain proteins that help the cancer to grow. Interleukin-2, or IL-2, is a standard treatment option. IL-2 helps the body's immune system fight cancer. Entinostat is a drug that inhibits a class of proteins called histone deacetylases, or HDACs. This study will help researchers learn if the addition of entinostat to high-dose interleukin-2 is better, the same, or worse than the usual approach to treating this type of cancer. IL-2 is approved by the U.S. Food and Drug Administration to treat metastatic melanoma and metastatic renal cell carcinoma. Entinostat has been given breakthrough therapy status by the FDA for advanced estrogen receptor-positive breast cancer. The combination of IL-2 and entinostat has not been approved to treat advanced kidney cancer and should be considered investigational. You may be eligible to participate in this study if you have kidney cancer that has spread to other parts of your body or cannot be surgically removed. There are other criteria that must be met to enroll in this study. Talk to your doctor to learn more. A full description of this clinical trial is available at www.clinicaltrials.gov using trial number 03501381.